that. So this will be uploaded into the YouTube channel for review later for anybody that wants to come back to it. And I'll also turn on the live transcripts, which you can turn off if you don't like them. Okay, so anyway, tonight we are back to our regular member finds like we were doing all last year, all the way up into January. I think people are starting to find mushrooms. I know I am. The way we work this is it's basically a show and tell. Show off the mushrooms either by sharing your own screen, or if you need to, you can email me the pictures of the mushrooms and I'll show them from my screen. Try to reserve ourselves like 3 to 5 species or so, maybe about 5 minutes a person, in order to let everyone get a chance to share, and everyone is welcome to share. And let's see what else. We usually get a queue going. So if you want to share from your own screen, please type your name into the chat box and I will just get a list going and we'll just call on people in the order that they have stuff to go. I know Dave asked me, I know Karen's here. Hi, Karen. Dave asked me if you could go fairly soon. So are you ready to go, Karen?
Yeah, I can if you want to. I mean, you all probably know more about these mushrooms than I do, so I don't really know what I can say about them. But
okay.
Yeah.
Well,
Go
I'll go with yours first, and then I'll get back into the regular queue.
Thanks.
Mm-hmm. So let's pick out 5 of these, okay?
Oh, Dave sent me. Shoot, now I can't see it.
Do you want to see the first email he sent me?
Yeah, he said he sent. I'm calling up his Mushroom Observer page. Come on, baby. Oh, Rhizocybe pruinosa, he's calling it. I don't know which one that is on your list.
Okay, I gotta find. He sent me a couple of emails. I'm not sure which one. He was excited.
Yeah, he's finding new mushrooms, so he's excited.
Here we go. This is.
I can go on to the next one if you want. That he sent me an email with 5 observations in it. So.
Oh, this is the first one he sent me. If this isn't it, maybe you can send the email to me. Oh yeah, this is it, right?
Yes, this is the one.
Yeah. Okay. So here's the name for everyone.
Let's see, by the 417. Actually, he must have. We were out looking for morels. I think he must have found these at the same time. I don't even remember.
Well, let's see what he has to say about them. So he was calling these, or somebody's calling these Rhizocybe pruinosa. He says here, I'm sorry, I'm looking for the notes. On woody debris, probably hardwood, the base of the stipe with long white rhizomes, spores 4.5 to 5.5 by 2 by 3, not amyloid. And it looks like there was a couple different names on here, but this pruinosa is one else. Let's see what they look like. Can you guys see that, or is it just a blue thing?
I'm just seeing a blue thing.
Oh, he's doing that.
Oh, there it is.
I've never seen that mushroom.
Yeah, I see he didn't even put down the location. I'm kind of surprised. So
maybe he didn't want us to see where he was at.
Well, I should have known, because I think, yeah, it had to be it's the same area where we picked morels.
Oh, well, you can tell us that. We won't. Well, a secret.
It's a place where we have picked black morels every spring.
That's why.
Yeah, that's why there's no location. All right, cool. Well, that's an interesting observation.
Yeah, I have nothing to add to what he wrote on Mushroom Observer.
Okay, so here's a black morel. Morchella angusticeps.
Yeah, this is from our secret undisclosed morel location, which has been producing very few morels ever since the ash trees died. But we did get a few there this year.
Yeah, unfortunately, I've seen that same thing here in that Philadelphia area.
Yeah.
All of our ash trees have died, and what used to be good spots are not really there anymore.
So it looked like there were white pine needles around them. Was it with ash trees or under white pine?
I guess there are a few white pines in that area. It's mostly tulip poplar, ash trees, birch, and the local understory trees, some oak. It's a very nice mixed forest.
Pretty. Thanks for sharing.
You're welcome.
Okay, so then we had Galerina marginata. It says here on mossy hardwood log.
Yeah, this is from our local club had a foray this past Sunday, and they found deadly Galerina, and Dave was happy to see that because he wanted people to know that this comes out in the spring and to be aware, learn what it looks like. He said they had some newbies, people who were coming to their first foray, and he wanted them to see this. So he was really glad that these things came out.
Yep. See the nice little tiny ring on there?
They will come out in the fall too. And they've eliminated Galerina autumnalis, I think, saying that it's a synonym for marginata, which tends to be the fall one. Anyway, they're poisonous in either case.
Was it growing on wood?
Yeah, usually it's a well-rotted wood. Can be on wood chips though too, and it can be by the hundreds and thousands.
Yeah, see, he said it was a mossy wood.
Yeah, I think it was on a log.
Did he make photos of the cystidia?
No, there was only the spores.
Oh, okay.
A mycena with no species name on it. I feel like we were just talking about that.
Yeah, if he doesn't have a species name, I got nothing.
So this is on a mossy log, probably hardwood, with spores 10 to 12 by 7 to 8 amyloid.
Yeah, this would be from the same foray.
We were just discussing before we started how difficult mycenas can be.
Did you happen to notice if they had any smell?
I didn't even see the specimens.
Yeah, a lot of times for people who aren't familiar with mycenas, the odor and the presence of latex or like a liquid inside of them are often help with identifying these little things. Okay, and the last one, tree ears. Labeled as auricularia. And possibly auricularia Angiospermarum on hardwood.
Yeah, these also came from our undisclosed Morel location.
Do you two eat these?
Yeah, he said, actually he didn't cook these, but he said he was looking for some because he said if he found them in good shape, he might put them into a stir fry.
Yeah, I was wondering if these are the mouse ear mushrooms that they might use in the Chinese cooking. Luke, do you know?
If they're not the same species, I never heard them called mouse ears, but I guess that makes sense because they're kind of fuzzy. But yeah, yes, they do use these or a very similar one in the same genus. They cultivate them.
Luke, what did you hear them called as in the Asian cuisine?
What do they call these? Wood ears.
Cloud
I
ears
think
sometimes.
cloud ears. Yeah, cloud ears is a name that I hear used sometimes.
Wood ears or tree ears is what Dave usually calls them.
Yeah, wood ears is I think what I usually hear them, but I've heard cloud ears as well.
My Asian friend calls them cloud ears.
Yeah, I think you don't want to eat too many of these if you're taking blood thinners.
Yeah.
Yeah, these are a known blood thinner.
Oh wow, never heard that.
Oh,
yeah,
yeah.
that's good to know.
Yeah, I had a friend of many years ago who was actually encouraged to eat these because as a natural blood thinner.
All right, awesome. Well, thank you for sharing, Karen.
All right.
If anybody wants to eat these things, they're available at Asian markets. I go to a Korean store in Cherry Hill called H Mart, and that's where I pick these up, either fresh or dry. And I've been really eating a lot of these in the last few months. I really like them. So my blood is as thin as it can be.
So Igor, if they're being sold fresh, does that mean they're being cultivated somewhere?
Yeah, they're cultivated probably in China and they're shipped probably like what, overnight or something. I don't know. Yeah,
Yeah,
they're in
that's
a
a
fresh
hell of a-
state, and you know, I cook them right away, and they have this wonderful texture, and they are very sweet, and I really love them. I love them more than inaki or shiitake or maitake or anything. I mean, I love these things, and I also been eating dry. I mean, you can pick up dried wood ear in a bag sealed, and they're not very expensive. And you know, you buy like 2.5 ounces, and it's good like for God knows how many servings. I mean, they really soak up the water, and they swell up and they get huge. So, but apparently they're packed with nutrients like all fungi. So, but I didn't know that they were blood thinners. That's something I should be aware of, just in case, because you don't want to have too much of a blood thinner in your system, I guess. So try
Cool.
them out. They're quite nice.
Yeah, and right now, obviously, as
good
you could
idea.
see from the stuff that Karen was sharing, they're outgrowing right now. I've seen some in the Philadelphia area. So they like this cool, wet weather.
Yeah, what we have is Angiospermarum and Americana. There was this paper that came out a few years ago describing these taxa from North America, but the original one called Auricularia Aurica judae actually is a European species, not found here. But they kind of all look the same.
Are they found on hardwoods, Igor?
The ones that I've seen always on hardwoods. I've never
Okay.
seen them growing on pines. Maybe one day.
Yeah, isn't that the distinction between Angiospermarum and what was the other species called?
Americana.
Americana. Isn't that the distinction? One's hardwood, the Angiospermarum is hardwood, and Americanum is the softwood.
Possibly, or maybe Americana grows on both kind of wood. So I have to check it out. Go to Michael Kuo's page. I think he has Americana on his website, but not the other one, not the Angiospermarum.
Mm-hmm.
I'm sorry, the auricularia that I see here in the Adirondacks is usually on balsam fir.
Balsam fir, really?
Balsam fir.
Yeah. Wow.
Yeah. And that's over, 10 or 11 years now.
Yeah, I remember reading in some of those papers, because Greg Thorne, the Canadian mycologist that came to speak to us a few years ago, he specializes in this, and he started talking to us about this. But I remember he was saying, and he recommended some papers that originally they had called it like the deciduous unit and the coniferous unit. And then, of course, with a little bit more study, they realized that those were actually two different species, and they separated them out. So, I don't know. They're not easy to work with, though. I've tried microscoping them, and I think you have to dry them first before you can get any sections out of them because they're so squishy. So, all right. So anyway, thank you very much again, Karen. And then let's get back into our list here. I think Lauren was the next person to volunteer. Would you like to share now, Lauren?
Hey.
Hello.
All right, we can just skip over this. I just wanted to show you guys what I saw this week, but we all know what this is, so we don't need to discuss.
That's the one. That's the species group.
Species group, right.
So I've been finding a lot of these, which I'm sure you all have, but I personally love the way these taste. But whenever I've been finding them lately, they haven't been from spots that are clean. So I just have been leaving all of them behind. Anyway.
Nova section by KCI. Yeah.
So this is now my second term. I found this last weekend, and so this was really exciting. This was right alongside a trail in Morris County. I still don't know much about Terramite. Well, I'm curious what you guys recommend for a source for learning more about this genus.
Are you on Facebook, Lauren?
Yeah.
There's a really good group called False Morels Demystified.
Oh yeah, I'm part of that.
Okay, they're a really good group. There's some good people in there, and there's not too much, racket and noise in there.
But I mean, like if I wanted to read literature about them.
Well, the Bassett's book, Bassett and Michael Bugh, their ASCO book.
Okay.
That's like the go-to ASCO book for North America right now, as far as I know. Somebody else might have another source too. But
what is it called?
It's I'll type it into the chat, or at least close to what it's called, and you'll be able to figure it out.
Luke, what was that group called? Was it False Morels Declassified? What was it?
False Morels Demystified.
Okay. Okay, thanks.
You've spelled Michael Bugh's name wrong.
Oh, sorry.
B-U.
I said it was going to be. Oh, you're right. That was
B-U-G, I think.
That was a typo. It wasn't even an accident. It's a typo. There you go. False Morels Demystified. I think that's what it's called.
Yeah, I need to do a little bit more learning. So this was fun. This is my second one ever. This I was super surprised to see. This is on my neighbor's stump right across the street. And I saw it from a distance because it was bright orange. So of course I went over and had to look. And I'm pretty sure it's Flammulina.
Lauren, we cannot hear you so well.
Oh, huh.
It sounds like the form. Goes and comes.
I'm like right in front of Mike. Is there anything I can do? I don't know. I'm right in front of this laptop. You're having a hard time hearing me?
Mm-hmm.
You're just fading in and out a little bit. It's not.
Okay.
That's weird. Okay. Anyway, Flammulina was days ago. This is the first time I found this in the spring, so this was fun.
Yeah, certainly looks like it.
Yeah, and like I said, I think I said white spore print.
Is this one?
Flammulina.
What?
Flammulina. I'll type it.
You don't
might-
have a picture of the bottom of the stem?
What's the genus?
Flammulina.
Oh, whoa, 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 I
Flammulina.
didn't hear that. Okay.
in my comment.
Flammulina granulata. Okay, I got it.
This is. All right. Well, I guess I'll end on that because for some reason my other pictures aren't showing up. But I wanted to ask you a question. I found two tiny little morels, which actually look, they were kind of similar to yours because I think they were attached. But I'm curious if you ever find morels on private property and they're really tiny, is there anything that you can do to protect them from wildlife eating them? Because I found these morels that were like an inch tall, and then I went back the next day and they were gone. I'm positive no people eat them. I'm positive that the deer or groundhog eat them. And I'm wondering, can you put like a cone over them? Will that stunt growth? Anything you can do for them?
I guess you could. Why not?
Have you ever
right?
tried that?
No, I would just keep that spot in mind and go back there some more.
Do you think that it would be bad if you did something like put a cone over it, like you need air to breathe or anything like that?
I don't know. I mean, you could give it a try. It sounds like a good experiment.
Is this on your property or somebody else's?
It might be my property, or it was my property until they were gone.
Well, I mean, on your property, so to speak.
It's my property. If you really must know.
Well, I don't live anywhere near you. I don't even know where you are. So
No, I know you're not gonna come.
Yeah, if it's on your property, especially, it's somewhere you'll be able to kind of monitor them every day. I mean, you could always play around and see if you could do something like that.
put a peach basket over it, sure.
Yeah.
Or you know what, just protect it. And if it's gonna get cold on Thursday, I would pick it before that because I've had frost damage things. I've found things like that that I've kind of tried to protect. And
They're
they
already gone. They
buy
were gone. Yeah, and they're no longer there. They were already eaten, so it's too late now.
But by who? Were they cupped stems that were left?
There was a tiny little chunk of a stem on the ground right next to piles of deer poop and deer hoof prints.
I can't
Yeah, I mean,
hear
we have wildlife that are back there.
you.
animals and slugs eat them too.
Yeah.
Well, yeah, I definitely think a deer ate them.
Ah,
It was a bummer.
Well, just keep an eye on that area. At least you know that they're growing in that area. So,
Yeah. Well, we get like 1 or 2 a year, so I think that was it. I will keep checking. All right. Well, thank you guys.
all right. Thanks, Lauren.
You're welcome.
All right, Brandon, I think you are next. Are you still there, Brandon? Brandon, you still there? If you're speaking, you're muted.
Oh, I'm sorry about that, guys. Yeah, sorry.
There you go.
All right. And am I sharing?
Not
Yes.
yet. Have you hit the little green button? There you go. It's
Yes.
coming.
I had to hit the final green button. Sorry about that. So I found a couple Ganoderma throughout the week, and I'm pretty new to this, so I just wanted to kind of nail down my identification of the Ganoderma genus. So I found a couple specimens on this substrate here, which the top was pretty deteriorated, so I wasn't sure, but I'm pretty sure that this is a fallen pine tree. I don't know if anybody's got any opinion on the substrate there. This is the underside of the one that I just showed you.
Are you
the
sure
one-
it's not a hemlock?
Yeah, I mean, it absolutely could be a hemlock.
Yeah, no, it doesn't have a stem though, does it?
It doesn't, and I got a cross section coming up. The cross section of that one. And I don't know, I'm calling this a suge. What do you guys think?
Yeah, if that's indeed a conifer tree, that's what we'll call that.
Yeah, I'm almost 100% it was a conifer tree. Suge will grow on obviously hemlock, but also other conifers, correct?
Correct.
I'm not so sure of that. They've broken up Ganoderma into a lot of different species.
Okay. Yeah, and
Um,
honestly, that's why I wanted to kind of present some of these. I did go back and took another.
The fact that it doesn't have a stem is a little worrying to me, but I don't.
So I think it has a little pseudo stem, but would suge be a little more pronounced in terms of the
Oh,
stem?
yeah. Oh, yeah. All right. That looks more like it could be. And definitely from last year's growth.
Yeah, yeah. Although this flesh wasn't too woody. This was actually kind of soft.
That's because it's probably wet.
Yeah, it was a little wet. But this is a little more like the coloration is pretty light on it. So I think if it is a pine tree, I be confident in suge there. Can I be confident in knowing that's a suge?
No.
Gotcha.
I would say, yeah, personally. There's only 5 Ganodermas that grow in New Jersey that I can name.
Okay.
Yeah, and I'd like to show you guys what I believe is a curtisii.
That Curtisii would be the other one that I would say what that would be, but Curtisii generally grows on hardwoods.
Right, and that's why I'm trying to nail down my substrates. I don't know how to identify a fallen, rotting tree. I'm having a tough time with live trees as it is without leaves right now. So these fallen, rotting ones, or the standing, rotting ones, I'm having a tough time with. But here's that substrate, and I'm thinking, that's a beech tree or a birch tree?
Not,
think-
I don't think.
I think many times if it's got natural bark on it, you could take a picture of it through picture this or, one of the apps that identifies plants, and you might get a good guess that way, but the bark would help identify it.
Right.
If it's lost its bark, it's kind of hard to tell.
Well, this bark up at the top is still present, and I'm only, again, my identification skills aren't great, but what other trees have that kind of lateral break in the, there are almost eyes forming throughout the top of this. And I guess the picture doesn't do it much justice, but I was noticing some eyes in real life.
Oh, everything else around it looks like hardwoods.
Yeah, so I'm at least pretty confident that this is a hardwood. Back to. So there's the specimen there that's got the more kind of elongated stipe. Here's a cross section of that one. Back to the first one. So I'm calling that Curtisii. What do you guys think?
Yeah, that would make sense.
One more that I'm a little more confident on. Is this what a dead hemlock looks like?
I don't know. I can't really comment on that myself.
Really have to look at the bark up close.
So I have, that's the best I would get there. These are the items that I found there. I guess, kind of the same issue that we'd be going through, either Curtisii or Suge, right?
I don't know. Let's see those other pictures again.
Of the substrate?
No, the ones on the ground. Do they have stems on them?
Yes. I think I have. So you can see the stem. This is the cross section and the stems down here.
Hmm.
Last thing, Curtisii grows like there's the lower part of the tree.
Yeah, right at the base.
And I have seen photos of the one on conifers, and it grows a little bit higher. I mean, if it is in conifers, yeah, that's different.
Gotcha.
You will know for sure, yeah.
All right, well, I guess I gotta just get better at identifying dead trees.
Are you close enough to go back in the summer?
Yeah. Oh yeah,
To
this
see if
is.
there's new growth. That would be well worth looking at.
Well, actually, you know what? I do have one photo I did want to share. Where it is now. So here are some new growth on what I was calling a birch tree that probably isn't. This is the one that I identified as a hardwood, and these were growing right underneath what I identified as the Curtisii.
Yeah, that would be right. If it's on hardwood and it's got those long stems on them.
Yeah, and these are obviously tiny. These are new growth for sure. And they're really shiny and really cool colors. The coloration doesn't really show here, but super purple and really yellow on the left.
So in New Jersey, you only have 3 of the Ganodermas that actually are orange and shiny, the lactate ones. That's Sugai, Curtisii, and Cecily or Sessile.
Okay. Cool. Well, that's all I got. And then, well, I have, yeah, that's all I'll share.
Okay, great. Thanks, Brandon.
Yep, take care.
Okay, so I am going to share my screen and go into some of these emails that I have received. Okay, so Marisol, you're next.
Okay. Oh man, have I made this bigger? Oh no. 1 second, my maximize. Okay, I got it. Okay. Make it bigger, please.
You want to see that name before it goes away?
Yeah, Galerina hypnorum. Okay, so that was on Sunday. I went to the porch in my house to like breathe some fresh air. Bless you. And then I had 2 pots.
One second, Marisol. If you're not speaking, can you mute yourself? Thank you. Okay, go ahead, Marisol.
Yeah. So I went to my porch, and then I turned my head to one side when I saw in one of the pots that I have hanging from the edge, this little mushroom. And I was like, what? And then when I saw it, I said, maybe it's a galerina, but I said, there is no wood there. And then I took it out. I made the sport print. I did the micro of it. The gills are forked in several places. It has a remnants of a veil. Yeah, you can see it in there photos are not that great, but that's the best I could do. And the cap is 2 centimeters in diameter. The stem is 2 centimeters long. This type is like a little wider towards the gills and a little narrower towards the base. And it's attached to the moss. So when I put the photo in iNaturalist, it gave me this name. And then that one grows in relation with moss. I didn't know that there were gallerinas that grew in that relationship with the moss. I did the sport print. It has beautiful cystidia, cystidia, or whatever name that is. And it's so, I did the side of the gills and I did the top of the gill. So the cystidia in the side of the gill is up to 100 micro units long. So I only saw one free. So when it enters to the flesh, it becomes a little wider and then it narrows into a like regular hyphal end. You will see a photo that is not so good. You can show more. Here are a few. That's the best I could do. This micro is not the best. Oh, another weird thing that I noticed is that many spores were, what's the right way of saying that? Instead of seeing them from the side, they were seen from one of the ends. So I saw them as circles. And there were so many of them like that. I have no idea why. Cystidia, top edge of gill. Oh, at the,
Sorry
yeah.
to interrupt. The reason you're seeing the spores in this projection is because there's a lot of space between the glass cover and the slide. You should really press on the cover to eliminate.
You know what happened? The spore print was thick.
Mm.
That makes sense. Okay, yeah.
But it's nice to see them in this particular arrangement because now you know that the spores are perfectly round. So
yep.
you can measure them in different in profile or in face view. You know, be accurate with your measurements.
It's called face. What's the name when you see this?
Well, there's profile and face view. Face view is when you see them either from the front or the back.
Okay.
And profile is from the side. When you see that picture to the side, that's profile.
Regularly you see from the side. Yeah. Oh, that makes sense. Cool.
And are these little crystals in there?
Yeah, plenty of crystals. And the cystidia on the top of the gills is shorter than the one in the sides of the gills. Yeah, you can show more. It was beautiful. I couldn't believe it. Cystidia, the top is shorter. That's on the top. It got some matter inside. And it's next to the basidia. I could not make a better photo here, but I measured the whole thing, focusing on different parts of the cystidia or whatever name that is. It measured 100 micro units long. And you see that at the center, at the middle of it, is like wider and then it narrows to towards the, it's inserted inside the flesh in the gills.
All right, very cool. So that's Galerina hypnorum.
Hypnorum. I wanted to know what was the meaning of hypnorum. I could not find it. I found the bumblebee, bombus hypnorum, and many other things are hypnorum, but I could not find the meaning of it. I wish I knew. And there was only one.
Very cool.
Mm-hmm. Okay, so a week ago, I went to my daughter's house, but I wanted to go to the woods. I have everything with me, and she didn't want to go, and so I could not resist, and I went to the backyard of the house where she's living. There were 3 sycamore maple trees, all huge, fat, and diseased. I mean, I'm sorry, I'm sick. I'm sorry, I meant to say sick. And I found many branches scattered on the ground, and I found like 19 different kinds of fungi on thick branches, medium-sized branches, and sticks, all from the same kind of tree. This the one I told you, sycamore maple. And so I found this Hypoxylon perforatum. It's the first time that I have seen it. It's beautiful. It's different to the other Hypoxylons because it's not black. And also the The osteols are white. So the contrast is pretty neat. I have seen it in the one book that I have here. And then when I posted in an iNaturalist and I wrote the name in there, then I check more photos and it looks like that. That's a good one. Very small. I did some of the, I tried to get the spores. They're dark and they have a slit that goes from one end to the other. Is that the right word, the slit?
Yeah, I think so.
Yeah, it's hard to see it because
Yeah.
the micro photo won't get bigger. Yeah. Oh, yeah, you can see it there better than in the other photo. I don't know the reason for that, but it's part of the clues for identifying some of these black ascomycetes. It almost looks like a coffee bean, kind of.
Yeah.
A little bit, yeah.
What book did you find this in?
In the same tree, I found this is the same wood, the sycamore maple.
No, what book?
Oh, in the book that from this Danish and French authors, there is 2 volumes. It's a new one. It came
Okay.
out 2020. You know what? I can't remember.
Okay, I was just curious.
Peterson and the French guy, I don't remember. It's there. Did you buy the books? Oh, I can't remember.
All right, Hypoxylon perforatum. Very cool.
And there are many observations in iNaturalist. I just didn't know. Okay, this one was found on the same wood that Acer pseudoplatanus. Oops. Oops. It's taking its time.
Yeah, let's try the next one. See if that loads.
Oopsies.
Oh, there we go. We'll go back to the other one.
Yeah. So this one is called Camellia tinctor. I never found it before, and I thought it was Diatripe stigma, because one time I remember I heard that when you remove the wood, but I'm sorry, you remove the ascus. I'm sorry, you remove the ascomycete, a little bit of the wood, you see red underneath, and it was red orange, but it wasn't Diatripe stigma. They corrected me, Ethan from New York Mycological Society, and they knew like at once because of the peculiar color on the wood. It really penetrates the wood for some good area. You can see in this one. What did you say?
That's really interesting. I've never seen that before.
Yeah, I didn't know this one. That was my first time.
So that red staining is enough to narrow that down?
Yeah, to tinctor.
Because it does show
Camellia.
diatribe. I would have
That
thought
was
that.
beautiful. Yep.
So now I'm gonna have to peel it back to look. Thank you.
Yeah.
Yeah.
Right. And I think that the edges, I was reading about this Diatribe, and then I read about this one. You have to pay attention to the way the edge is, because in some of them the edge is raised, and in some of them is flat to the level of the wood. But even looking underneath is important too. And the color is like red orange. It's really pretty.
What kind of wood, please?
The sycamore maple.
Oh, right.
Yeah, all of these things were in, yeah, except for the gallerina. Okay, this one was the tiniest stick you could imagine, smaller than the pinkish. And I saw these little things, and the photo is not that good. So it looks like I made a drawing, the second photo, I made a drawing of it.
Okay, everyone have that name?
Riti, this, oh boy. Riti hysteron rufulum.
There you go.
The second photo is good. It's made out of like two lips.
Yeah.
And the lips have like, oh gosh, how do you say that? No smooth, like wrinkled.
You're talking about cracks.
No, each lip has some wrinkles. You show them, in the lower photo, on the lower towards the left.
Right
You
there.
can hardly see, yeah. I made a drawing to show that better because my photos couldn't be better than that. Because it's so tiny too. Okay, so it's something like that. It's made out of two lips, but what happened is that it was rolled in because it was kind of dry. And inside the hymenium, yeah, the hymenium, the one that I color black, it has the most beautiful magenta wine color. But I could not show that in my photos when I found out what it was. And then I made a cut, and you can see what it is. It's like it's kind of a cap. And the hymenium surface is very small. It's so tiny, 2 millimeters long. I made the micro so I can see the spores. The funny thing is that, no, oh boy, that's still wrong.
Okay,
What
we'll skip
happened
that.
there?
Is that right?
I think so. So the spores are segmented. Yeah, you can see that some have 2 parts and some have 4 parts, 4 segments. Oh, so I couldn't do better job than this. Those are the paraphyses, which are infertile structures that accompany the ascus, which is the fertile part. Can you show the ascus look on the lower part of the picture? Lower right.
Oh,
It's
right here.
very submerged, yeah. And what you see on the left, like red brick color, is the pigments that are at the end of the paraphysis. There is like a layer. What you see on the top as the color of the cap. All right, so now you can see the spores. I do not know why the asci were so far down. As you can see, the distance between the tip of the ascus and the edge with the red pigments. And some of the asci have 5, 6, 8 spores. It was irregular. Some of the spores didn't have any segments. Maybe they were younger. But none of them have more than 4. This is, yeah, photo of the asci with this colorful material on the edge. Yeah.
Very cool.
So I asked Eric Boehm, he's the expert in these things, and he told me that's the one, Rufum. He told me the species.
So these will be in that group they call hysteroid fungi?
Yeah.
Because they have that slit in there, right?
I don't know if the type of the spores, because when I went online to look for photos, people wet them for a long time and then they open.
No, the fruiting body itself, because it has that slit in there, and right when they get wet, they open up and expose the hymenium. And Dorothy Smullen, she was telling me that they call that group the hysteroid fungi,
Okay, okay, yeah, yeah.
which you can see is kind of that name, Hysteron is in that name.
There, yeah, yeah. Rhytidihysterum. And there are a few species of this Rhytidysterum. I didn't even know that because that's the first time I ever found it.
Very cool.
Right. And on the same type of wood, I found these super tiny black caps. Super tiny. I don't have a better photo than that. I think that the biggest, I can't remember, it was like 1 millimeter maybe, or even less. And the wood was pretty, it was decorticated, very dry. And it's like a troops of them. And the spores have several segments. So you can see that some of them have drops and then other ones have these segments. I don't understand, I never found them before. And the paraphyses, which are the infertile parts, were beautiful. They were branched and had this black blue matter at the end. So you can see in that one, I show a photo of the ascus and some of the spores. 1, 2, 3, 4, 5, 6, 7, 8 parts the spores have. They were huge. Sometimes it's so funny because the smallest thing you find, the spores are so huge. 8 spores.
Good picture.
Per ascus, yeah.
So for anybody that's trying to decipher this, this is the ascus, right? Is that right? Ascus with all the spores inside of it.
Some of the spores and some of the ascus there.
All right. So that is Petiolaria atrata.
And it's funny that some spores, as they mature, they divide in few kind of fungi, not all of them. Okay, so last one, Ethan helped me with the names for several of those. This is the tiniest thing ever. It was 2 millimeters wide and it's made out of several spheres, black spheres. Yeah. So imagine this, that thing is 2 millimeters wide. And he told me that the name of the ascomycete, and he also told me it could be the asexual stage of the name that he proposed, and it was, and I made the micro. So this one is really odd because in the ascus, there are hundreds and hundreds of spores. Here is before they are just starting to come out of the black balls. I press the ball in the slide and the things showed up. Okay, and then you can show the next photo.
Wait, I'm sorry, I had a question. You said that's an ascus and those are hundreds of spores in there?
Yeah, yeah. It's one of the few that have, okay, now you can see the ascus came out a little bit more and you can see the amount of spores inside.
Wow. And you said this is asexual or sexual?
Oh, I messed up. This is the regular. It's because I found so many that I'm confused now. This is the regular. It's the ascomycete, the sexual stage. I'm sorry, I messed up. It's right. You're right. Thank you for the correction. Sorry.
That's crazy. With all those spores in there.
There are like few. Okay, here I press the slide and they just went out of the black ball. They shoot out. And then they came, the ascus fell apart and these things went all over the place. I don't know if anybody ever tried to count how many spores are there because it's too many.
That one's really cool.
Nischia brumeana.
All right. Thank you, Marisol.
All right, thanks.
Okay, looks like John and Nina are next.
Yeah. Okay.
So this first one is this, the mix.
Yeah. Yeah, that's the next day, putting it outside, and John had a picture of it fresh in the field too. It's we found it in the, oh, I didn't find it. John and Jed and Lizzie found it, and it was found in a fire break, and it's a fire mushroom. It comes up with a fire. It's pine, those pine trees, and it's amyloid, and let's see what I can say about it.
What's
Oh,
the name?
sorry, it's Mixomphalia M.
Mixomphalina.
Mura.
Okay.
It's the one you found
Yeah, I remember. Yep.
in the fire pit. Yeah, same thing.
Yep. Cool.
It likes the fire,
Mhm.
and but it has amyloid spores, and it's not very amphiloid. It's goes down a little bit, but not it's slightly decurrent maybe, but not very much.
And we were walking along a trail, which was actually a fire break. And the one side of the trail had a fire maybe a year or two ago.
Oh, anyway, that's that one. And these are some of our precious Mycena, the pain in the neck things. A whole lot of these black ones. I got two pictures of black ones, and they all got different spores. So I guess the only thing I got to do is just to go back and put a label on them so I can look them up. Sorry, Liz. She has the same. Yeah, so like I was saying before, there's some of these have like circular, the spores are round. Some of them are oval with like roughened. Some are not roughened, and it just and the different sizes too. But these all in sphagnum moss. So I figured they were all the same, but they weren't.
No, I think there's a lot of undescribed Mycena.
Yeah. Yeah, you think?
Yeah.
Okay. Well, that's.
And they were all in close proximity.
Yeah. Okay, so Jed took some nice pictures of stuff, but he put a lot of stuff on Facebook from the walk we took, and I tried to contact him, but I guess he didn't get my email to put some of the stuff on. Next, yeah, this is John's picture of it in the field.
That's the Mixo.
Yeah.
Paulina.
Yeah, it's kind of a waxy, kind of moist feel. This is it. Yeah.
Sorry, I can't seem to make this picture any smaller.
Oh dear. John. Okay, then it's okay. I just sent you his email.
There's that one. Okay.
Okay.
Then you had this one.
What else did I have? Oh yeah. That's the white one of those things, the Neoflavius. But you said that was an Americana now, the one with the white?
So yeah, see the white one. So
Yeah.
There was a paper that came out last year where a group of Chinese researchers, they had one collection of this from Connecticut, and they described a new species called Americanus. There's a white one. But the problem, I guess, with that name, from what I'm hearing, is that there was only a single collection made of it, and they described a whole new species from a single collection. So people are kind of iffy about that name.
Okay.
And with the sequencing that's been being done, we're starting to see that there may be a bunch of different species out there that are all really similar looking.
Okay. So
I think the name Americanus is kind of being people are hesitating to use it. I know I was using it a little while back, but now I'm kind of seeing the logic of what people are saying.
But Luke, if they have the DNA that anchors the species epithet, unless you have several species having the same DNA.
Right, you have a valid point there. But okay, you're right. You have a valid point there, but I guess we need to look at the DNA of the stuff that's around here to see if we're looking at the same species that they're looking at in
Right.
Connecticut.
You're saying it could be more than one white species, and you have to distinguish them either microscopically or by the molecules.
Mhm.
So you know, anytime you find a white Neophthalus, we have to sequence it and see if it matches or if it doesn't match. I wouldn't be surprised if we have more than one species.
Yes, so we just sent in 3 or 4 of them to
I
the,
know, I processed them, so still waiting.
and I've been collecting even the orange ones. We're starting to think that maybe there's a group of them as well, the ones we,
Right.
what, Avularis. I think that's
Yes.
kind of being referred to as a group now too.
So should we save those? Because I find those all the time.
Yeah, I think we should be. I think we should be saving them, but as long as we're making good collections, like we need to really be looking at the spores and
Okay.
getting dimensions and getting good photographs.
Yeah, by the way, Mycoflora is bowing out of sequencing for identification. They will no longer support any projects after April 30th. Their focus is on conservation, I think, now. So if you find something rare or unusual, they'll sequence it for you. But if you just want to ID fungi through internal transcribed spacer, ITS, that's a no-go. They will post a list of vendors who do sequencing later on. That's what they say. So we have to find somebody else eventually.
Good.
All right.
Okay.
So
Next week we might have some lichens to show because we have this lichen thing at Franklin Park, which Lizzie and Dorothy and Jason are going to be and what was the name of it? Dennis is it? Are going to be showing telling some people about lichens and
Oh,
how
great.
to identify them. So if we find something interesting, we'll put it on Zoom.
Dennis, is it Dennis Waters?
Yes,
Yes.
yes.
Oh, great. He knows lichens.
Yeah.
He did a survey of all the lichens of Hunterdon County, I think,
Mm-hmm.
and published it through the Philadelphia Botanical Club.
Yeah.
All right. So you are up.
Right, well, I'm going to bore you with my usual thing about my sarcosomas, but first you can look at a beautiful little yellow tremella that I just photographed yesterday. I assume it's mesenterica. It's on alder. I forgot to check whether the alder is dead or alive, but anyway, I was kind of surprised to see it. We had been dry up until last week, but this past 4 or 5 days, we've had quite a lot of rain. So I just thought it was pretty, and I was very surprised to see it at all, to tell you the truth. And then my next bunch is the little sarcosomas. Right, this particular one was taken today, and that's about 3.5, 4 inches across. This is becoming what they're looking like. They're still sitting up. They've also all plumped up over the last few days, but they're also starting to open out, to flatten. And I expect by next week, I may be able to blow across them and see spores.
Susan, how big do they get?
This one could get a little bit bigger, but that's about the biggest they get.
Oh, okay. Like 4 inches?
4 inches across is about
Okay.
the biggest they get.
Cool.
And between last week and this week, okay, now this picture, you can see the snow. This picture was taken on March 30th, okay? You can see how nice and little compare, those are Norway spruce cones. And then the next picture, that was taken today of the same exact mushroom. So that's 3 weeks apart. And that's what they do. That one was probably a little bit frozen, but already coming up. And see that little one, I almost missed that little one underneath it that had come up even just since last week. But that's what they do, is they sort of swell up and then kind of wrinkle. And then that black disc will flatten out. And by next week, I expect there'll be spores. So that'll be four weeks that it's growing.
Is that a northern species? Because I never see that down in central.
It seems to be associated with Norway spruce. It seems to be circumboreal, if that's the right term, Igor. I don't.
Yes, it is.
Seems to be common in Northern Europe and in Russia. I haven't investigated further than that. There is also a tiny little, almost colibia-like thing, sort of at 2 o'clock next to it on the right. I have a name for that, but I haven't really investigated what it. The other things are so much more interesting. Yeah, I have to get a better picture of them, but I saw them at each one of my 4 sites today. But look at the skin on that thing. It's just so weird. Isn't that cool? And then, like I said, I'm anxious to see how much that little one underneath it grows. Okay, then I have another.
Nice photograph, Susan.
So Susan, what's the small fungi that are growing on the cones?
Oh, I don't think it's growing on the cones. It's growing on the ground because there's quite a few of them around. I'll have to look up the name, and I'm sure they'll still be next week. Paula DeSanto gave me a name. Actually, maybe it's on the cones. You know, I have to look. I didn't look at that until I saw the picture, Igor.
Looks like there's some on both sides of the mushroom. Here at this cone that you're showing. Luke,
Yeah, it actually does look like it might be on the cone.
and there is some on the opposite side.
Yeah.
9
yeah.
o'clock.
I don't know what that is.
They're tiny.
Oh, yeah.
Oh, yeah,
Yeah.
that one actually shows it on that. Isn't that funny?
They
Now,
are definitely coming out of the cone.
Yes.
I'll just say my eyesight isn't so good. And by the way, I have to sort of get down and use, you know, I have kind of a bifocal lens, eyeglasses, and to get down low enough to take the picture, I can't always see the right whatever. And the camera does a better shot than my eyes do. And it's just a moto, you know, phone.
So are they Biospora?
Oh, I don't think so.
Biospora Myosaur?
Yeah, all right. Somebody's put up a name on the cone, Strobelarius esculentus. Now that, yeah, I think that's the name that Paula gave me, and I did not associate it with the cones, but that it could very likely be with the cones.
Yeah, the estrobilo, whatever, that's the one on the cones.
Okay.
There are few
What
species.
that is.
But Biospora are specifically growing spruces too.
Huh.
So I know that you're right. There are a couple different ones.
Well, and this is very early for anything to be out. So I should investigate more as far as look at them more carefully and even bring them home. I do have a microscope, but I'm lazy about it. But you know, I've never looked at the spores of the sarcosoma, so I should try and do that when I know they're sporing.
We want to see them, please.
Yeah. Oh, you're
all
gonna
of
make me
these-
get out my microscope, huh?
You have to finish. You gotta go to the end.
God, I haven't used the microscope in ages, but I have one. I do have a really good one. Jeff and I bought it about 30 years ago. And
I forgot. Those sarcosomas, are they ascos or are they?
yes, yes. And the spores form on those black circles. Now this little group here, or at least 3 in a row here, this was taken on April 6th of this little fruit body in my site 4. This was taken last week on the 14th of the same exact mushroom. It was very dry for about 10 days. So most of the little mushrooms I photographed last week had shriveled up or just plain didn't grow. but I couldn't believe, and I'm sure it's the exact same place because of the location of the tree. And, you know, I sort of go through the woods, through these sites in the same general direction kind of thing. And then look what it looked like today. There it is. It's plumped up again. And the little black disc where the ascos are going to form, Igor, by next week or the week after for this little one. My site 4 is a little slower than the other couple sites because it's a little more protected, remote, colder. Yeah, and this is another one that the animals have nibbled on. They seem to nibble just on the black disc part mostly. And you can see at about 1 o'clock, there's a little bit of the black disc that they left behind, but the rest of it is the case or the main body of the fruiting body with the jello, or I call it a little jello shot. And they seem to really like to eat the black disc part off. Although a couple that I had last week, they're entirely gone. They've been taken away by the animals entirely, whether it's red squirrels or deer or whoever. Both are very prevalent in that woods. But I just look at the texture of that skin though. Isn't it cool?
Yeah.
So that's all I have for this week. This was also taken today.
Great. Thank you, Susan.
So basically these sarcosomas, they hang around for a while and they're not in a hurry to grow very much.
No. The first year I watched them for 9 weeks.
Wow.
And they literally will eventually dry up into a little brown potato chip. I mean, literally, they look like a little brown potato. I mean, they shrink up to nothing. They're so fun to watch. And what's interesting is the moss and stuff will grow around them. So by the time they get to a potato chip, there's a little bowl left in the moss where they had been. Very interesting. Anyway, that's it.
Do they regrow in the same spot or they move around?
They grow in the same 10 to 20 foot area, depending on which site I'm at.
But not from the same actual spot. So you had a mushroom in 1 year at a certain location spot, and then it regrows in the same spot next year, no?
No, correct. Yeah, they tend to move around a little bit. And I've noticed that I keep looking at holes that I think are from sort of last year. And yet, you know, there's a lot of animal activity and I don't even know what, but this year there seems to be more holes in the moss kind of thing, but some of them could be from holes from last year. And I also have noticed that in, especially my site number 2, that the animals have really nibbled and taken some completely away. They're just gone entirely. One I had a really good, 2 I had really good pictures of. I wanted to see them further, you know, along in their life, but they've been eaten. So anyway. It's a lot of fun to watch. Good excuse to get in the woods too.
Yeah, well, save a collection for us. We'll put in our herbarium, unless we already have it.
I would be very surprised if you have it, because when I sent some to Jean Yetter at the Botanic Gardens in New York about 6 or 8 years ago, the Botanic Gardens did not have them at all. And if you go to their website, the picture that's there is mine from about, well, the picture's probably from about 9 years ago. Best picture I took, but yeah. And you know, Igor, the only way you can dry them, because there's so much, they're all water, is I have to put them in a wax bag, put them in the back of my, you know, sort of fold it up, put it in the back of my refrigerator and forget about it for about 2 or 3 months.
Yeah, that's what I do with morels, because I don't dry them in the dryer because they emit so many spores, it just clogs everything up.
Right.
And I just leave them on a towel for like a week, and they just dry naturally in the fridge.
Yeah, that would work.
Yeah.
I will try and collect one when I'm sure that they're making their spores though, because now I know they're too early.
All right. Thank you, Susan.
Uh-huh.
All right. Thanks, Susan.
Mm-hmm.
All right. I have 5 here. First 3 are morels. Different species of morels.
I wish.
So this is Angusticeps. All of these were found either in Philadelphia or in Bucks County, Pennsylvania. So I found this one. This is the nicest black morel I've ever found. I usually don't find black morels hardly ever. Or, you know, I'll only find 1 or 2 at a time. And this one was just all by itself, and it was actually right on the street in Philadelphia, right at the edge of the woods. I was actually not even looking for morels. I was parking and getting out to go into the woods, and literally I got out of my car and looked in the grassy strip on the side of the road, and there it was.
So Luke, is it under oak or some kind of hardwood?
It's all hardwood there: tulip poplars, oak, and beech.
Or disturbed ground if somebody's been digging up something. Foundation, bulldozing, whatever.
Yeah, maybe. This is a really high traffic area. A lot of partying going on back there. So,
Could be.
yeah. So anyway, this is a black morel, Angusta ceps, and really showing off its namesake. And then I just started finding these the other day, the diminutivas. So these are the really small tulip poplar morels that I find specifically under tulip poplars trees. And this thing was this was a big one, about 3 inches tall. So these are really small.
Like a big thumb.
Like a big thumb, exactly.
Before you even said name and tulip, tulip poplar, that I knew it was a tulip poplar morel.
Yeah, they have that very specific shape to them, don't they?
They do.
They're
They
very
and
slender and long.
And the stem is kind of smoother than some.
Mhm.
They get pretty big later on. I mean, I found them and they're like 4 or 5 inches tall, but they're the same, that long, skinny, more fragile than
a good year, or there's some other kind of tree they might be growing with. I think I rarely do you ever see them more than about 4 inches.
Yeah, I picked some last year in this area that were pretty big, and I was surprised at how big they were. But you're right; usually they're pretty darn small. So,
Quite nice.
and then finally this one. So I'm pretty sure this is Americana. But look at this; it's a two-headed morel.
Nice.
And that I actually found under an ash tree, the rare ash tree that I can still find this alive. So this one actually looks like it's been out for a little bit. Just look how, it's already kind of browning on there. That might be frost damage, right?
You still ate it though, right?
Yeah, oh yeah, I came home. For sure.
Hey, Luke, do you think if you cut them, they grow like that? Because it's private property. It's not mine. It's a farm of a friend. And I cut them and then I'll go back a week later. And sometimes out of that same place, there'll be a new morel.
Really?
Yeah. I try not to pull them out of the ground.
Right. Arlene Bassett was just saying that maybe morels are one that probably should be cut because they form sclerotium underground, which I'll be honest with you, I don't even really understand what the sclerotia do. I thought it was like a nutrient storage vessel.
Yeah.
Mm-hmm.
But Arlene Bassett was speculating that the other day.
Yeah, I just don't want all that dirt in the basket, and I might as well leave it undisturbed. I just had a little pair of scissors and I cut them for a knife.
Years ago, when we used to hunt morels in Warren and Sussex County, you could start about the end of April and go over the same site about every week to 10 days and find more morels if it was a good season. I mean, literally, 10 to 20 to 30 more in the same exact spot. So it depends on the weather a lot too.
Mm-hmm. Okay, this next one, this neat little thing, Foliotina rugosa. I usually find this underneath of logs around here. And this one got a little bit messed up when I messed with it because it has this really distinct ring on it, this white spot. It has a little tiny ring on it. And this is a really little tiny fragile mushroom. It's only 3 inches tall, maybe, and super thin. Although they do get a little bit bigger. I've seen them a little bit bigger than that. And they tend to be growing out of like this really rotting wood. But these actually have mycotoxins in them, or amatoxins, I'm sorry. So it used to be a canosoby. Oh. So Foliotina rugosa.
What was the old name?
Canosoby?
Oh, conocibe rugosa.
I guess. I think I just saw that the genus was once called.
Does he have a veil, little ring thing in there?
Yep, that little white thing
Oh,
right there.
yeah, yeah, yeah,
That's
yeah.
a little veil.
Oh yeah.
It's actually pretty distinct, but it's so delicate
Oh, yeah.
that I think I touched it and it just flaked right off.
How big is the cap?
Maybe a centimeter across.
Oh.
Really tiny.
Yeah, and very, very long.
Mhm.
The
Nice.
old name Wikipedia says is Canosoby filaris.
Okay.
Filaris makes sense.
Yeah.
Like a thread, the stem.
Mm-hmm.
Yep, and well known to have amatoxins. So
Hemotoxins.
That was underneath the log?
that's underneath the log. That's how I usually find them.
So you flipped the log over and that was there?
Yep.
Ah,
How tall?
Maybe 3 inches.
Okay.
What'd you say, Igor? Hemotoxins? Because I
No,
thought
I think amatoxins. Yeah, the same as phalloides and Galerina marginata. Yeah.
Yep.
Cyclic cyclic peptides. Yeah.
So one of the really deadly mushrooms.
Don't eat those.
Don't eat them. Okay, and last but not least, I collected this little fella, New Fabulous Avularis. At least that's what we're calling that, right? So this one was really nice and fresh. So I did collect this, and I got a spore print, and I dried it. So they call these the hexagonal pored polypores. I guess when they get a little bit bigger, they get a little more of a hexagon type pore shape to them. But I did get spores out of this one. These things seem to give up spores pretty easily.
What kind of spores are those? Boring?
Yes, kind of ellipsoid. I think 4x11 ballpark. Smooth, yeah. Not much to them. Like little beans, little clear bleak beans. It is a pretty mushroom, though. I think.
Yeah, nice color on that one.
Yeah. All right, that's my 5. And then,
Good job, nice showing.
thanks. Liz.
and this is more little mycena. And we found several different kinds. This was in the cedar swamp. And the pictures don't do it justice. It was actually darker than that. It keyed out perfectly in Rene Leboeuf's mycena key to megaspora. It all looked perfect until I measured the spores and it really didn't match at all.
They're not that mega, huh?
They're not that mega, no. Kind of ellipsoid, but actually it was a darker, much darker than that, but little bell-shaped cap. The stem itself was blacker toward the base than it was up at the apex. And I kept it separate. So, but Nina's probably right. We should go back and just number our finds because we were in the hardwood swamp with sphagnum and also in the cedar swamp. But probably multiple kinds, a lot of different looking spores, but not megaspora.
Does anybody ever try to make use of some of those European mycena books? I know that there's that mycena site from Norway or somewhere that's got just tons and tons of European mycenas on there.
Is that online?
Mm-hmm.
Well, this was amyloid. It didn't bleed. It didn't have any smell. So, other than the description, which was darker. I'll have to look at that.
Yeah, I wonder how many of those are actually circumboreal because they're saprobes.
Yeah.
None of the ones we picked had any smell.
No, none of them. And none of them had any latex.
No.
Cute little things, though.
Yeah.
And it was nice to find gilled mushrooms for a change.
Yeah, for sure.
Thank you.
Okay, thanks, Liz.
Thanks, Liz.
Okay, that was all of the all the observations that I had saved up. So is there anybody else that has anything they would like to share? No? Crickets?
I
All
have
right.
a couple. I can do some if you don't mind.
No, we don't mind at all. This is what it's for.
Okay, all right, here we go. Let's see. Let me go into, this is from today, and I think it was the 12th. So I'll start with the 12th. Let me get to that open. Oh, I have to close this again. That one. Okay, here we go. All right, so this nice blue color ring, can you see this? Everybody okay?
Yes.
Yeah. Great. Nice colored ring, and I'll show you that's the underneath of it. Let me see if yeah. So the piece of bark is actually sticking out, and you can see more of the bottom than you can see the top. So I don't know, it's gorgeous, but I don't know what it is.
Is it really that blue?
Yes, it was really quite like a deep jean color.
And that's on a conifer.
That's what it looks like to me, yes.
Heterobasidium.
Blue like that?
Heterobasidium.
Right. You think that's heterobasidium being that blue?
I don't know if they are so blue, but it fools you looking like an ostrea. And then when you look underneath, you see the pores, and it grows at the base of conifers. And it smells so good.
I mean, you're right. The shape is right.
I never remember to smell them. Darn it.
Yeah, yeah. Nina, what do you think? No, it's not there.
I've never seen a blue like that. I have no.
Not so blue, but I remember I have found it 2 times within 2 or 3 months, and it fooled me because I thought it was Stereon, the one that we call Ostrea. Yeah, it's pretty blue.
Yeah.
But the zonation is what reminds me of the Heterobasidium.
So you should put on Mushroom Observer. Maybe some people have some ideas about it.
Oh, I'm so new, I'm not even on Mushroom Observer, sorry.
Are you on Facebook, Bianca?
Yes, I'm on that mushroom group on Facebook. I
Cross
forget.
fungi and polypores?
Yeah, I was going to suggest that too. Crust fungi and polypores. That's a group that's pretty good with this stuff.
Oh, okay, I don't think I'm on that one. All right, so if you don't mind putting it in chat, I'll be able to copy it. So then I'll go to the next one. So there's this little tiny one you can see is compared to my hand. Also on a conifer, you can see the cones on it, and I have a zoom in. Let me see if I can get even closer. They look like these little dots, but they do have a stem, little orange stem. Let's see if I can bring it closer to this.
Yeah, that's going to be a Dacromyces or a Dacropinix type thing.
Right.
A little jelly.
Mm-hmm.
Grow out a little further,
Gotcha.
but they are beautiful.
Thank you. All right, that was from about a week ago. Oh, this was actually a shock to me. I was expecting to find it to be a polypore, but then when I turned it over, it was a gorgeous vein-like gill look.
Rametes betulina.
Yeah, it is a polypore. It's just long pores. It's an evolutionary in between, I call it.
Oh,
I
wow.
learned it as Lenzites betulina. Is that What is it now, guys?
Trametes. Trametes.
Termetes betulina?
Oh, okay.
Probably having been on birch, but maybe not.
Yeah, there were birch there, so that makes sense.
Mhm.
All
But
right.
it grows in different types of woods too.
Okay.
Yeah, yeah.
Oh,
Alright.
oh, you're thinking in Pictoporus betulinus? The one that only grows on birch? This one grows in different types of woods and is also betulinus.
Okay. Gill polypore.
That's new to me, so great. So this is today. We were walking, this is Durant Park in Freehold. And there's, I think we did a very similar, we did the lesson about these, and I think you can take them off the tree and carve into them, if this is the right one I'm thinking of. So let me,
Another one? Oh no, this is, do you know the tree?
it looked like
Black locust?
yes, black
Okay,
locust.
so this is
Yeah.
the Robinii, the Fomitiporia Robinii. Yeah,
So
the black locust. Yeah, yeah.
there you go.
Yeah.
Yep. And there's a bunch of them. So there's, I have at least 15 pictures, but I don't have to go through all of them. So these are all, this is the same as this one. This is the same as, oh no, this is a different one. All right, so this was actually quite fascinating to me. One was growing from the other. So it's here. So I have several different perspectives. This is horizontal as you're looking at, the tree had fallen. So you can see in the background, there's the vertical tree. So the tree that this mushroom is on, it has fallen. And then now I'm looking down towards the ground, again, a perspective. So one is growing from the other one. It's very interesting.
What I know about this one is that when the tree is standing, it's alive, it's a parasite. And when the tree falls down, then it reaccommodates and grows as a saprobic.
Oh,
First it was a parasite, and now when the tree is down, it's a saprobic.
cool.
So it could grow, it keeps growing from the old fruiting body. Yep.
That's
Yeah.
what's going on here. That's really cool.
And the other thing is it always wants to face its porous surface,
Down.
bearing surface down.
Uh,
Tree changed direction, the fruiting body had to change direction. It's called, I think, geotropism, right?
Yep.
Wow, that's a thing. Wow. All right.
That's unusual for Pholinus to do that. Has Pholinus got a new genus name?
Yes.
This one does.
Yeah, what is it now, Luke?
Fomitiporia or something like that. Fomitopsis or Fomitiporia. Look.
Wait a minute.
Oh, you're looking?
I'm looking because I can't
Oh,
remember.
sorry, okay.
I'm so impressed that you can remember these names.
It's in all your books as Pholinus though. So look at that, Pholinus Robinii. And almost all the locust trees around me have it growing.
Oh, yes. So here's another one that's also very similar. It had the one direction and now it's the other way.
Yeah,
So,
yeah, that's a really good picture of that. Yeah.
and this is enormous. Here, I have a picture with my hand. I think this is the underside, and there you go. That's my hand next to it. This is bigger than my hand with the bulbous size. Let me go back to the previous.
I found one and I measured it. It was 43 centimeters long, the longest side. Yeah.
Wow. Yeah, that
Then,
makes
and
sense.
then it was like, I visited several times, and then we had a foray there in Rancocas Nature Center, and I told everybody, please do not cut the polypores, and then you just toss it outside. So nobody cut it, and then I went back months later, somebody took
Aw.
43 centimeters big. I was so like, no.
Oh,
There
man.
are many more. It's very common in my area.
Yeah, so this is, again, I had it. There's
Beautiful.
another one quite large.
Oh, yeah.
Did you happen to feel the undersurface to feel if it's putting out a new layer of tubes?
A new layer of tubes, no, but I did make an X on it to see if it stained, and it did
Well, it
not.
would be kind of moist feeling if it's putting out a new layer of tubes.
Well, this sort of has kind of like a cut in it. Oh, it's not a great picture. Sorry about that. But with this, they all
That's-
were. Sorry, go ahead.
That's more like a slug or some insect damage, looks like to me.
Okay. Let's see.
I put the new name in the chat.
Yeah, that's good. Yeah, let me get that.
So here's again a bunch of them just growing all over this tree. I couldn't reach it to see get a better photo, but they were quite many. Here's another one. This one looked darker underneath. Maybe is it older because it's darker?
Yeah, probably.
Okay.
Or you have to go around and feel them to see if they're still alive. If they're dead, they're dry. I mean, they're really just dry. But if they're kind of moist and sort of like a dog's nose, they're
Okay.
still alive. They still have food and can grow out a new layer. And
Okay.
now is when they would be doing that. They would be starting to do their new growth for the season. See,
Okay. Yeah.
those concentric semicircles is a new year's growth
Oh, I see.
repeated underneath with pores.
I see. Okay. I'll pay
That's
attention
really-
to that.
You can see that looks like it's at least 15 or 20 years old, that.
Susan, I have to say something about that. That was the general assumption that each zone of growth meant 1 year, but it's not. Yeah, now they explain it. They say that it could be seasonal growth. It could be that the weather was more moist or whatever, so it grew one more layer, but it doesn't mean necessarily that is 1 year.
Okay.
Did anybody
Okay.
hear that too?
Okay, so it's 1 season's worth of growth.
Correct. Yeah, I've always heard that too,
Yeah.
Marisol, that like it's just a good growing
Yeah.
time. A nice wet period, then it cools off and dries
Well,
out,
that's
and
it.
it
Thank
gets
you
wet
so much.
again.
I'm gonna get all the names from the chat. So I hope you guys put them in order. So I have, let's see. So this blue one, this orange one, this green fuzzy, it was a soft one, which apparently it's a polypore with gills. I'm impressed again. And then the one on the black locust. All right, that's it. Thank you.
Thanks,
All right,
Brianna.
thank you. Thanks for sharing. Okay, anybody else? All right. Well, everybody get to bed so you can get up at the crack of dawn and get out there and look for the morels before you have to go to work.
Wait, I'm missing some names.
Well, I think the first one, Marisol suggested, I'll type the first one in there that she suggested. So that first one, that blue polypore, Marisol had suggested that. But I would definitely put that up on the that Facebook group, and it definitely has that form, but the color looks really different. That's why I was asking about the color, because I've never seen anything that color before.
It's funny, Luke, because when I was at one of the fungus fests, I bought a magnet from one of the artists that had this picture of a blue polyspore like this. And I'm not at home to take a picture of it, but maybe I'll put it on Facebook where it has it on the magnet because I thought it was so pretty.
Ah. Okay. There you go. You got all those names now, Bianca?
She's missing the lenzites,
Oh. All right.
or whatever it's called now. Lenzites betulina is what I learned it as. The gill.
Betulina or us?
Yeah, that's another thing. I'm not good with Latin to remember which.
There you go. That's close enough.
So Tremidas betulina is the
That's the guild polypore.
one, and the fluvis. Oh gosh.
The Fulvofomes robinii. That's that big gigantic one
That's
that
the
was on
big
the
thing.
black locust.
Okay.
The Dacromyces, that was those little
The
yellow
potato-
jelly ones.
Myces. Okay, Gabromyces and the Lubiniae. Got it. Great, thank you.
All right, you're welcome. All right, well, thank you everyone that shared tonight, and I guess we'll see everybody next week, right? All right,
Thank you.
have a good night, everyone.
Thank you.
Thank you, good night.
Good night.
Good
Good
night.
night.
Bye-bye.